Everyone, thanks for attending another one of Universal Sequencing's Q&A sessions. As Aaron mentioned and alluded to, we've had a pretty big uptick in people new to Universal Sequencing's technology show interest in this forum. So I'm going to take the first 15 or 20 minutes to give a very quick overview of the technology, as well as a handful of representative applications. Hopefully this will be a good learning experience and or refresher. If you feel like you are fairly familiar with universal sequencing as a technology, now would be a good time to get a coffee, because I'll be just going through some slides pretty quickly over the next 10 to 15 minutes. So I'm Andrew Anfora. I am the Senior Field Application Scientist Manager. I'll be doing the bulk of the presentation. We have Aaron Levy, who's our Chief Commercial Officer, also present. David Nordell is our Inside Sales Manager. He's dialed in. Certainly not last but not least, Tom Chen is our Chief Scientific Officer. Didn't have his name listed on this particular slide, but he will be here for answering questions as well. So I'm just going to launch right into it. Feel free to type questions into the chat as you think of them. We're going to go through the presentation first, and then we'll address questions. David will be monitoring the chat. David, if you see like a lot of questions on a given slide, feel free to interrupt me. Otherwise, I'll just blow through this.
Will do.
All right. So just to summarize what we're going to be talking about, Universal Sequencing produces TELL Seq, WGS Library Prep Kit. This is a simple, fast, one-tube library prep assay for long-read sequencing. It converts short-read sequencer data into highly accurate long-read sequencer data. And we can capture molecules greater than 100 kb in length. Relative to most library preps out there, especially in the long read space, we are a very low input assay, requiring sub-nanogram to nanogram quantities of DNA. And I'm going to explain the biochemistry of our library prep, as well as cover some applications, including de novo genome assembly, a preview of our metagenomics analysis pipeline, go over some examples of structural variation, whole genome phasing, and example of targeted phasing of exomes. So to hit on our technology, this diagram should largely be at least 2/3 review for everybody in attendance. What we have is a representation of different flavors of next generation sequencing, where we have a diploid organism represented with one chromosome colored blue, the other chromosome colored red. With short read sequencing, you get highly accurate short reads of 100 to 500 base pair that are also relatively low cost to generate. The challenge with unlinked short reads is it is nearly impossible to phase haplotypes, certainly if they are further than 500 base pairs apart. It's really difficult to confidently say that two given variants are present in the same chromosome. Additionally, de novo assemblies are quite limited and can't achieve contigs of significant length if you're only using short reads. One way around this is to use long read sequencers such as PacBio and Oxford Nanopore. These sequencers can generate more longer read lengths depending on the instrument and the run mode, looking at highly accurate reads of 10 to 15 kb. In the case of PacBio, you do have the option of running in a lower accuracy mode to get slightly longer length reads. And the tens of kb read lengths do allow for some degree of phasing of a genome and a reasonably robust de novo assembly. A linked read solution such as TELL Seq is taking short reads, but prior to fragmentation will be applying a UMI to the intact long molecules such that the library fragments can be traced back to a parent molecule, and we can therefore link the reads, preserving a lot of that large molecule information with the high base calling accuracy of a short read sequencer. And we are able to capture molecule information upwards of 100 kb, and I'll have a couple of bits of data later on supporting that claim. I'm going to go very quickly over our mechanism of action. I don't want to spend too much time on this, but here is how we actually generate our libraries. Really an important thing with our process is we don't have any exotic capital equipment associated with our workflow. Everything takes place in PCR tubes and a magnetic stand. If you're already running NGS library preps, you won't need any exotic equipment to start using TELL Seq. What we do is, working from left to right on this diagram, we start with mixing a transpososome complex and our TELL beads. The TELL beads each contain a unique 18 base pair barcode. So we have a potential barcode library of roughly 2 billion sequences. So we mix the beads and the transpososomes, and then we add high molecular weight DNA. Ideally, we want to be loading DNA molecules 40 kb and above. You can still get some usable data out of TELL Seq with molecules as small as 10 kb. So depending on how you are preparing your DNA samples may already be fine, or you may need to do some minor adjustments to isolation to really maximize the data coming out of a TELL Seq assay. So once the beads, transpososomes, and DNA are mixed, they will form a complex to which we add a second transpososome for tagmentation. We will break and wash the DNA, and then we generate amplicons using sequences on the two transpososomes. So the subset of your long DNA fragments that contain the proper orientation of the transpososomes will be amplified into Illumina compatible libraries containing your P5 and P7 tags. This is a very fast workflow. From isolated DNA to library ready for sequencing takes only 3 hours. So it would be possible to go from biomass isolation to initiation of collecting data all within a single workday. I'm not going to spend a lot of time on this. There's actually a fair bit of detail here. I do want to mention that we provide analysis software for free, either as downloadable Docker or Singularity images, and some of our tools are currently available on BaseSpace, and we are going to continue to be adding our software tools to BaseSpace for those that prefer doing their analysis on BaseSpace. So all of our universal sequencing provided software is depicted in green on this chart. Our data type are very similar to 10x Genomics legacy linked read offering. So we do also provide a conversion tool that converts our 18 base pair indexed format into a 16 base pair indexed format that is compatible with 10x Genomics tools. So if you're used to using Long Ranger or Supernova for linked reads, you can continue to do that. And not shown on this diagram, after running our QC tool TellRead, you can take those FASTQ files and do some third option not shown. So again, just to summarize the functionality of the assay, it is a 3-hour library prep using very low DNA input. So sub-nanogram quantities for microbial and targeted panels, and up to 5 nanograms for larger genomes. We're able to capture super long fragments and preserve size information for molecules greater than 100 kb in length. So really, even amongst assays that are doing some type of read linkage, we are capturing much longer molecules than pretty much anything out there. And our software is free. Just for comparison sakes of the DNA input, just a reminder, so when we're talking about 0.1 to 5 nanograms of input, if you're using a actual long read sequencer, you'll be needing microgram quantities of DNA. So we've had some users be able to enjoy using TELL Seq because they're able to do DNA isolations from a single insect and still have enough DNA to create that single organism de novo genome assembly. So now I want to quickly go through some data examples before opening things up to questions. I'm not going to spend a lot of time on this because it's available online. We have collaborated with Illumina on several fronts, and one collaboration was in microbial de novo genome assembly. And this is an application note available on the Illumina website where it was describing the utility of TELL Seq as well as the availability of some of our software tools on BaseSpace. Very briefly, we looked at a wide range of bacterial genomes, including high and low GC organisms. So we are able to handle high GC organisms. What you can see on the chart to the right is higher GC organisms will have slightly lower library yields. Just we are using a PCR step to amplify our libraries. So as you'd expect, slightly less yield with the very high GCs. I also like this study because we were DNA fragment sizes of 20 kilobases were used. So that's actually smaller than optimal, but they were still able to get really great data. This is a very busy chart. I'll just say the top part is describing the genomes that were used. So it's the reference genome information. The middle part of the chart is looking at the actual data collected. Illumina was collecting 100x coverage. I believe that is what they need to do if you're assembling a unlinked genome. For our workflow, we typically recommend starting at 50 to 60x coverage, and that's often sufficient for a high quality assembly. This data set had, as I mentioned, molecules of approximately 20 kb in length in populating the data sets. When we say SLF there in the middle two rows, that's an abbreviation for super long fragment. And that is the computed fragment length calculated by our software. And in the case of these microbial genomes, we were able to assemble the majority of the genome, the majority of the chromosomes into single contigs. We were also able to assemble the majority of plasmids for the organisms that contained plasmids, and we're able to produce data sets covering typically 99% of or greater of the genome. The worst we did with in this study was 97.28%. So we're really able to assemble near as no difference the entire bacterial genome. So yeah, single contigs. We were able to de novo assemble plasmids. So if you have total DNA isolation containing plasmid, you'll be able to assemble the plasmids into contigs as well. And just to briefly contrast what a microbial genome assembly looks like with TELL Seq compared to a Oxford Nanopore dataset and unlinked data with Illumina. You can see that there is that tremendous difference in DNA input, just night and day, less than a nanogram versus a microgram for the other options. We are assembling the genome into a single contag, so largest alignment and largest NA50, of course. Since we're using the Illumina base calling, we have no misassemblies, very few mismatches per 100 kb, and very few artificial indel artifacts in the data set. So really a quite robust single microbial genome solution. So we are so good at single microbial genomes, we have started to look at doing metagenomic samples. This is a tool that will be forthcoming. So very briefly, if you have a microbial community sample, you would follow the same initial steps of our workflow. So you'd still prep the library, you'd still run our QC tool, and you'd still run our de novo assembly tool. We are bringing online a third software tool for this workflow that will assign strain level identification and quantify abundance. So we've been testing this with some of the ATCC microbial community mixes. In this case, it's a summary of a 20 species staggered mix, where represents a community with pretty high dynamic range of species abundance. So far, we have been able to handle the 3 most abundant groups of microbes quite well, able to assemble and assign identity to all of the more abundant species. The species that are present at a fraction of a percent, we're able to identify their presence, but coverage becomes limiting for the de novo assembly. So we're still building out our guidance for recommended dynamic range and really what best practices would be and what you could reasonably expect for your microbial community sample. So we're very close to having a tech note out, and we have been presenting a poster with some of these data at a few conferences. Going to move quickly on to phasing of human genomes. Really quickly, just going to say that most of our human genome work is in 12878 and 24385, the genome in a bottle samples, as you would expect. Our data sets typically have the overwhelming majority of molecules being greater than 20 kb in length, with a significant portion of the molecules being greater than 100 kb in length, and that really lets us do some of these robust phasing and structural variant calling analyses. We have really good recall and precision rate for both SNPs and indels. As you'd expect, a little bit harder to call indels than SNPs, just like any other platform. Not going to spend too much time on these figures, but these data are from our genome research paper, which is really a great comprehensive description of what TELL Seq can accomplish. Just briefly, we were able to phase the MHC region, and here is a depiction of the variant assignment for some of the class 1 HLA and class 2 HLA genes. The regions where you can see a miscoloration or what appears to be an incorrect assignment are in regions of lower confidence in those references. So these are tricky genes to phase, and likely some of these assignments of being called classes incorrect are likely as much a reflection of the reference as it is this data set. Additionally, just another example of why it's important to phase, what you can see. Here's another region, and this is also in our genome research paper, where we have a two deletions of different size in the different chromosomes in the same region. So we had one 19 kb deletion in the first haplotype, and the second haplotype has a 114 kb deletion. You could imagine if you weren't able to robustly resolve the haplotypes, you would probably only be able to detect that 19 kb deletion and just think you had some low coverage for the remaining 100 kb of that 114 kb deletion. Yeah, so we can phase diploid genomes well, even in the tricky regions. Just wanted to briefly show the relationship between DNA molecule size and observed phase block. Similar trend would hold true for our de novo assembly applications. So what you can see here is the exponential gain in phase block size relative to input molecule length. So it's really been data observations such as these that lead us to the recommendation of using DNA input molecules, ideally 40 kb in length or greater. Depending on the specific project need, you still may be able to get a contiguous enough data set for your need with molecules shorter than 40 kb. Generally, we recommend that you use at least 10 kb DNA fragments into the library prep. We'd kind of expect some pretty diminishing returns after that. So again, even if you can't get 40 kb DNA molecules, you still may see some benefit from TELL Seq. Molecules greater than 40 kb are going to give you the best resultant data. So just to go over a couple examples of scaffolding really quick. So in the de novo assembly space, a lot of users already have pretty good PacBio data sets or pretty good Oxford Nanopore data sets. We've found that our ability to capture those 100 kb molecules, as well as having a different biochemistry of library preparation and having different breakpoints in our assembly, there's still really good value in using TELL Seq to further scaffold an existing genome assembly. So here's an example. Here's two examples of, again, a pretty good PacBio assembly, a pretty good Oxford Nanopore assembly that was made more contiguous by adding TELL Seq data to the assembly. And of course, you could also just use TELL Seq for a de novo assembly as well. I'm leaving out examples of that just for the sake of time. Yeah, I already said that. I just want to close with an example that we are also able to phase exomes. So we have a protocol for phasing or for using TELL Seq with SureSelect. Essentially, you run our TELL Seq protocol with some extra PCR cycles to ensure there's enough product. And then you go right into the Agilent SureSelect workflow. The only modification needed is using blockers that we provide rather than the standard SureSelect blockers. And we're then able to phase the majority of the exome. And again, we have a protocol for using TELL Seq with SureSelect that's available on our website. Yeah, so we're able to phase exomes. We've also done some other enrichment technologies that I'm not showing for the sake of time, and just want to close by saying that we have offices in California and Massachusetts, but we are supporting the world.